Hello everyone, and today we'll be starting with the new session of the topic photosynthesis in higher plants, and we'll be studying about the dark reaction in detail today. And within the dark reaction, we will be studying about the C3 and the C4 cycle. What are the biochemical reactions involved in these cycles? What is the significance of both of these reactions? Okay, so let's start with the dark reaction. So dark reaction is also known as the biosynthetic phase. Okay, and why it is called biosynthetic phase? Because there is a synthesis of the carbohydrate molecules using the product of the light reaction. Okay, so this dark reaction or dark phase has also been given the name of Kelvin cycle and C3 cycle. And one point here to remember is that the reactions going on in the dark phase of photosynthesis are light independent. Means they doesn't require light. Only the product of the light reaction that happens before the dark reaction are utilized in the dark reaction. Okay, so what are the products of light reaction? Those were ATP and NADPH. Both of them are used in the dark reaction or the dark phase of photosynthesis, but light directly is not involved. So that's why all the reactions are light independent. Now let's study the C3 cycle in detail. So the C3 cycle is the most common pathway for carbon dioxide fixation in plants. Okay, and how can we define C3 cycle? C3 cycle is a series of biochemical reactions that are taking place in the stroma of the chloroplast. Okay, so what is stroma? Stroma, we all know stroma is the colorless fluid present inside the chloroplast. So this is the site of dark reaction. Okay, so what happens in C3 cycle? In C3 cycle, what happens? The atmospheric carbon dioxide is converted into organic molecules, which are your carbohydrates, and primarily glucose is synthesized. Okay, so the major product is glucose, and glucose can further be converted into other carbohydrates such as starch and cellulose depending on the requirement of the plant. And both the starch and cellulose are utilized as a structural building material by the plant. Okay, this I have already told you that the products of the light reaction, okay, the products of the light reaction, which are ATP and NADPH, are used for the synthesis of carbohydrate in the dark reaction. Now another point here to remember is the enzyme involved. This is the most important point. The enzyme involved in C3 cycle is rubisco. Okay, rubisco, the full form is rubulose 1,5-bisphosphate carboxylase oxygenase. Okay, this is the key enzyme involved in the Kelvin cycle or C3 cycle. So it has two activities. Rubisco enzyme has two activities. First is the carboxylase, second one is the oxygenase. What does this mean? Carboxylase means it can combine with carbon dioxide, whereas oxygenase activities means rubisco can bind to oxygen molecule, leading to the formation of some different product, and that process is known as photorespiration, and we'll be studying about this photorespiration in next session. Okay, today in this session we are concerned about the carboxylase activity of the rubisco enzyme. So what does this rubisco do? Rubisco acts as a biological catalyst, and it catalyzes the reaction between the carbon dioxide and a 5-carbon compound known as ribulose 1,5-bisphosphate. Okay, so this is the compound. So rubisco catalyzes the reaction between carbon dioxide and a 5-carbon compound RUBP, leading to the formation of the 6-carbon compound, which is initially unstable, and it quickly dissipates into a 3-carbon compound. Two molecules of 3-carbon compound known as 3-phosphoglyceric acid. Now, what are the different phases involved in the Kelvin cycle? So here we'll be studying about Kelvin cycle in detail. Okay, so the first phase involved is the carboxylation phase. First one. In carboxylation, what happens? The atmospheric carbon dioxide, okay, which is in the inorganic form, is converted into organic form. in carboxylation phase, means carbon dioxide is being fixed. Now second phase is known as the reduction phase. Okay, and reduction phase is also known as the glycolytic reversal phase, means it is the reverse of glycolysis. Third phase is the regeneration phase, where what happens, there is a regeneration of the RUBP molecule. And this phase is very essential because it ensures the continuity of the Kelvin cycle. We'll be studying about all these phases now in detail. And some of the examples of the plant where this Kelvin cycle occurs is rice, wheat, oats and rye. Now let's start with the first phase known as the carboxylation phase. So in carboxylation phase, what happens? The carbon dioxide molecules combine with a 5-carbon compound known as RUBP to initially to generate a unstable 6-carbon compound. Okay, so initially a 6-carbon compound is produced that is quite unstable. So what happens? This unstable compound quickly dissipates into a 2 molecules of 3-carbon compound known as 3-phosphoglyceric acid. Here you can see from 6-carbon compound a 3-carbon compound is produced known as 3-phosphoglyceric acid. That's why Kelvin cycle has been given the name C3 cycle, because the first stable product formed is a 3-carbon compound known as 3-phosphoglyceric acid. Here you can see here in this biochemical reaction, carbon dioxide molecule here combines with RUBP in the presence of the enzyme Rubisco. Okay, it forms 3-phosphoglyceric acid. Okay, so now why I have written here 6 molecules of carbon dioxide and 6 molecules of RUBP? Because we all know that glucose consists of 6 atoms of carbon. Okay, it is C6H12O6, means it has 6 carbon atoms. Okay, so 6 carbon dioxide molecules are required. 6 carbon dioxide molecules are required for the synthesis of 1 molecule of glucose. That's why we are writing here 6 molecule of carbon dioxide. So if 6 molecules of carbon dioxide should bind to 6 molecule of RUBP, okay, in the presence of the enzyme Rubisco, which acts as a biological catalyst, it will give rise to 12 molecule of a 3-carbon compound known as 3-phosphoglyceric acid. Okay, why 12 molecules we are saying? Because 3-phosphoglyceric acid is a 3-carbon molecule. Now second phase is the reduction phase, which is also known as the glycolytic reversal. Okay, so in this stage, basically 2 biochemical reaction takes place. Okay, first one is the conversion of the 3-phosphoglyceric acid that were produced in the carboxylation phase to 1,3-bisphosphoglyceric acid. Here you can see in this reaction 3-phosphoglyceric acid, 12 molecules were synthesized. Okay, so 12 molecules of 3-phosphoglyceric acid utilize 12 molecules of ATP to form 12 molecules of 1,3-bisphosphoglyceric acid, and 12 ATP are produced. So point here to remember is that first one is 3-phosphoglyceric acid is converted into 1,3-biphosphoglyceric acid and ATP is being utilized here. And you have to remember 12 ATP are being utilized here because there were 12 molecules of 3-phosphoglyceric acid. Now the second biochemical reaction that takes place in the reduction phase is this one. Okay, in this reaction what happens? There is a conversion of 1,3-bisphosphoglyceric acid that were produced into 3-phosphoglyceraldehyde. And the enzyme here involved is a dehydrogenase enzyme which utilize NADPH2 and it converts NADPH2 into NADP+. Here again there were 12 molecules of biphosphoglyceric acid. So 12 molecules of 3-phosphoglyceric acid glyceraldehyde are produced and 12 NADPH2 are utilized. Okay, so after the formation of 3-phosphoglyceraldehyde, what happens? Here you can see 12 molecules are synthesized. Out of these 12 molecules, 2 molecules are, okay, 2 molecules of 3-phosphoglyceraldehyde are converted into glucose. Okay, so all these reactions are reverse of glycolysis. In glycolysis, what happens? Glucose is broken down into smaller molecules. Okay, and finally pyruvic acid is produced. Here, what is happening? 3-phosphoglyceraldehyde, okay, follows a pathway, okay, which is the reversal of glycolysis, and glucose is finally produced. Now, third phase is the regeneration phase. In regeneration phase, what happens? The remaining 10 molecules of 3-phosphoglyceraldehyde are recycled back to regenerate 6 molecules of RUBP here. Ribulose 1,5-bisphosphate, which is nothing but your RUBP. Okay, so this stage is also very important because it ensures the continuation of the cycle. If no RUBP is produced, the Calvin cycle will stop. Okay, and there will be no production of the sugar molecules. And again, another point to remember here is that here 6 ATP are used for the synthesis of 6 molecules of RUBP. Here, this you have to remember, 6 ATP are being used here. Now we have studied about the biochemical reactions that occur in the C3 cycle. Okay, now what is the input? What goes in the C3 cycle? Okay, and what is the output? What comes out of the C3 cycle? What are the products of the C3 cycle? Okay, so 6 carbon dioxide are being used and 1 molecule of glucose is being synthesized. 18 ATP were used, okay, for the conversion of initially 12 ATP were used for the conversion of 3-phosphoglyceric acid into 1,3-bisphosphoglyceric acid, and then again 6 ATP were used for the generation of 6 molecules of RUBP. So in total we have 12 initially were used, and then later 6 were used for the synthesis of RUBP. 12 were used for the synthesis of 1,3-bisphosphoglyceric acid. For the 1,3-bisphosphoglyceric acid, yeah. Okay, so in total 18 ATP are used. Here 18 ADP are produced, and 12 NADPH were used for the synthesis of 3-phosphoglyceraldehyde, and 12 NADP are produced as a product. So this is the output of the C3 cycle. Now this is the flowchart of the C3 cycle. So what is happening in C3 cycle? There are 3 phases. First one is the carboxylation phase. Second is the reduction phase. Third is the regeneration phase. Carboxylation phase, what happens? Carbon dioxide, okay, combines with water initially. Okay, so before combining directly to ribulose 1,5-bisphosphate, carbon dioxide first combines with water to form bicarbonate ion, okay, which is HCO3. Okay, this is bicarbonate. This is known as bicarbonate. Okay, and this bicarbonate then reacts with RUBP, ribulose 1,5-bisphosphate. Okay, leading to the formation of a 3-carbon compound. This is a 3-carbon compound known as 3-phosphoglycerate or 3-phosphoglyceric acid. Now, 3-phosphoglyceric acid is reduced, okay, to form 3-phosphoglyceraldehyde. Here we can say 3-phosphoglyceraldehyde is produced, which is again a 3-carbon compound, okay, and 3-phosphoglyceric acid is further can be converted into, you can say, glucose, sucrose, starch, depending upon the requirement of the plant. Now, some are utilized for the synthesis of the sugar, and remaining of the 3-phosphoglyceraldehyde, okay, they go into regeneration step and they regenerate RUBP, and in this process ATP is used, okay, and ATP is converted into ADP. So this is the entire schematic of the biochemical reaction going on in the Kelvin cycle or the SATHEE cycle. Point here to remember is first phase is carboxylation phase, second is reduction, third is the regeneration phase. Okay, and you have to just remember what are the reactions going on in these three phases. And then you have to remember what goes in the C3 cycle and what goes out of the C3 cycle. Now, I've told you that the reduction phase of the C3 cycle is known as the glycolytic reversal. Means reverse of glycolysis. So let's see how the reduction phase of C3 cycle is reverse of glycolysis. So first we have to talk about glycolysis first. So in glycolysis what happens? ATP and NADPH are synthesized in glycolysis. Whereas in the reduction phase of the C3 cycle, ATP and NADPH are utilized for the synthesis of what? For the synthesis of sugars such as glucose, sucrose, whatever, and some other carbohydrates, starch and cellulose. Okay, and what happens in glycolysis? The glucose is broken down into smaller molecules. Okay, so it is a catabolic process. Catabolic process, what happens? There is a breakdown of larger molecules into smaller molecules. Okay, that's why glycolysis is called a catabolic process. Whereas in the reduction phase of C3 cycle, what happens? Smaller molecules are initially synthesized, which are 3-carbon compound. Okay, and then they combine to form a 6-carbon molecule, which is known as glucose. Glucose is C6H12O6. Okay, that's why it is called an anabolic process. So here clearly you can see the reduction phase of C3 cycle is the reverse of glycolysis. Okay, so here we can see this is the steps involved in glycolysis. You can see glycolysis, what is happening? It is a catabolic process. Glucose, okay, follow this pathway, and finally pyruvic acid is synthesized. This is the breakdown of the glycolysis. So what happens in the reduction phase? It is the reverse of glycolysis. So what is happening in the reduction phase? 3-phosphoglyceraldehyde is being produced. Okay, and then reverse is being followed here. Okay, 3-phosphoglyceric acid is converted into 1,3-bisphosphoglyceric acid, and then finally 3-phosphoglyceraldehyde is produced. 3-phosphoglyceraldehyde here, okay, is further converted into glucose. It is following this pathway. Glycerylate 3-phosphate converted into fructose 1,6-bisphosphate, and then fructose 6-phosphate, and then finally to glucose. Okay, so here you can see reduction phase of C3 cycle is the reverse of glycolysis. Now let's talk about some of the main points of the C3 cycle. The first point here to remember is that C3 cycle is the fundamental process, means it is the essential process of the photosynthesis, and it is the most common pathway of carbon dioxide fixation present in the plants. Okay, so this C3 cycle is responsible for capturing and converting the atmospheric carbon dioxide into organic molecules such as carbohydrates, okay, or sugars you can say, and mostly or majorly glucose is produced as a major sugar in the C3 cycle, which is used as the energy source by the plant. Okay, so now what happens in certain conditions such as high temperature and excess oxygen? So what happens? C3 cycle in these conditions is less efficient than the usual. Okay, so these conditions, if present high temperature and excess oxygen, or you can also say high light intensity also, then C3 cycle becomes less efficient. Okay, and this is due to the occurrence of a phenomena or the cycle known as photorespiration. We'll talk about this photorespiration cycle in next session when we will study about the oxygenase activity of rubisco. But here you have to remember that in high temperature, in high light intensity, and in excess oxygen condition, C3 cycle becomes less efficient. Now, what is the significance of the C3 cycle? First one, it assists in plant growth. Okay, so what happens here? The glucose and the other carbohydrate molecules that are produced, okay, by the various metabolic reactions, okay, they are they help the plant in the cell growth, in cell repair and reproduction. So they assist in the plant growth. Second one is that the C3 cycle supports life on Earth. How it supports life on Earth? It supports life on Earth by providing energy in the form of organic matter. And on this organic matter, the heterotrophic organism, they feed on this organic matter, and this is how the life is sustained. Now coming to the C4 cycle. C4 cycle is also known as hatch and slack pathway. Now we have already I've told you that C3 cycle in certain condition is less efficient. So what happened? In plants that are residing in tropical areas or in arid regions, you can say semi-arid regions, they have some adaptation. Okay, and that adaptation of the C3 cycle is known as the C4 cycle. So here I have written that C4 cycle is the alternative photosynthetic pathway, alternative to C3 cycle. Okay, and where it happens? It happens in the plants which reside in dry tropical regions or in semi-arid regions. So we can say it is an adaptation to this dry regions or semi-arid regions. So now C4 cycle is an adaptation that allow the plants residing in the tropical regions to overcome the inefficiencies of the C3 cycle under high temperature and intense light conditions. Another point, very important point here to remember here, is that carbon dioxide fixation takes place 2 times in C4 cycle. In C3 cycle, there was carbon dioxide fixation was only 1 time with the help of the enzyme Rubisco. Here, carbon dioxide fixation takes place twice. Okay, one is by Rubisco enzyme. One enzyme involved here is Rubisco. Okay, and second enzyme here involved is, we will study about this enzyme, PEP carboxylase. This enzyme is known as PEP carboxylase, phosphoenol pyruvate carboxylase. So some of the examples where C4 cycle is seen, okay, so usually it is seen in the members of the monocots, and you can see some of the members of the Poaceae family, okay, like sugarcane and maize, and it is also seen in the members of Cyperaceae, in the members of the family Cyperaceae. Here are the examples: sorghum, bajra, panicum. C4 cycle is also seen in some of the dicots. For example, Euphorbia, Amaranthus, Chenopodium, Boravia, and Atriplix. Now, C4 plants show a typical leaf anatomy. Okay, so this is a kind of adaptation to semi-arid regions, and this leaf specialized leaf anatomy is known as Cran's anatomy. So what happens in Cran's anatomy? There are 2 types of photosynthetic cells. 2 types of photosynthetic cell. First one is the mesophyll cell. First one is the mesophyll cell. Second one are the bundle sheath cell. Bundle sheath cells. Okay, so here in this diagram you can see there are 2 types. For the first one is the mesophyll cell. Second one is the bundle sheath cells. And both these photosynthetic cells, they form a concentric ring around the vascular bundle. So here, inner side, you can see in the center, you can see the vascular bundle, and both the mesophyll cell and the bundle sheath cells, they form a concentric ring around the vascular bundles. Or you can say the bundle sheath cells and the mesophyll cells are arranged in a wreath-like manner around the vascular bundle. Now, what happens in mesophyll cells? There is a enzyme present which is known as PEP carboxylase here. Okay, so in C4 cycle, two enzymes are involved, rubisco and PEP carboxylase. PEP carboxylase, phosphoenol pyruvate carboxylase. Okay, and in bundle sheath cells, another enzyme is present, which we have already talked about, rubisco. So what happens? Rubisco, now you already know, combines carbon dioxide with RUBP. This is the role of rubisco. It combines carbon dioxide with RUBP, leading the formation of 3-carbon compound, 3-phosphoglyceric acid. Whereas PEP carboxylate, what it does? It combines carbon dioxide with phosphoenol pyruvate, leading to the formation of a 4-carbon compound known as oxaloacetic acid. Okay, which is a 4-carbon compound. So that's why it has been given the name C4 cycle, because the first stable compound formed here is a 4-carbon compound, which is known as oxaloacetic acid. Whereas in C3, the first stable compound is a 3-carbon compound known as 3-phosphoglyceric acid. One more point to remember about Cran's anatomy is there are two types of chloroplasts are present. Okay, so dimorphic chloroplasts are present. In bundle sheath cells, agranal and large chloroplasts are present, whereas in mesophyll cell, granal and small chloroplasts are present. Now let's study about the steps involved in the C4 cycle. Now, first step is the initial carbon dioxide fixation, or we can also say as first carbon dioxide fixation. I've already told you that carbon dioxide fixation takes place twice. So this is the first carbon dioxide fixation that takes place in the mesophyll cells. Okay, so these are the mesophyll cells. And in mesophyll cell, which enzyme is present? In mesophyll cell, PEP carboxylase is present, which combines carbon dioxide with phosphoenol pyruvate, leading to the formation of a 4-carbon compound, oxaloacetate, or you can say oxaloacetic acid. Okay, so here you can see what happens. Carbon dioxide initially combines with water. You can write here water. Leading to the formation of bicarbonate ion. Bicarbonate ion then combines with phosphoenol pyruvate, okay, and leading to the formation of a 4-carbon compound known as oxaloacetic acid, which is the first stable compound. Now, what is the second step? In second step, what happens? There is a transport of this 4-carbon compound to the bundle sheath cells. Okay, the oxaloacetic acid is converted first into malate. Okay, so this is how it is converted. Oxaloacetic acid is not directly transported to bundle sheath cells, but oxaloacetic acid is first converted into malate, or you can say malic acid, and then it is transported to the bundle sheath cells. Oxaloacetic acid is converted to malate here in mesophyll cells, and then this malate is transported to the bundle sheath cells. These are the bundle sheath cells. So what happens when the malate reaches bundle sheath cells? The bundle sheath cells, there is a decarboxylation of this malate. Okay, means removal of the carbon. Okay, decarboxylation. And what happens? This decarboxylation of malate results in the formation of a 3-carbon compound, pyruvate, and carbon dioxide is released in the process. Here you can see in this diagram, malate is decarboxylated and a 3-carbon compound, pyruvic acid, is produced, or you can say pyruvate. Carbon dioxide is released during this process, which enters the Kelvin cycle. Okay, and the carbon dioxide is fixed to form the sugar molecule, which is glucose. So what happens in the bundle sheath cells? Malate is transported to the bundle sheath cells. Malate is decarboxylated to form pyruvate, and in the process carbon dioxide is released. Carbon dioxide enters the Calvin cycle. So here second carbon dioxide fixation takes place. This is the second carbon dioxide fixation. Second carbon dioxide fixation takes place in the bundle sheath cells. First carbon dioxide fixation takes place in the mesophyll cell. This is the second carbon dioxide fixation that takes place in the bundle sheath cells with the help of the enzyme Rubisco. Now, what is the significance of the C4 cycle? First significance is that it helps in the reduction of a wasteful process which is known as photorespiration. I've already mentioned photorespiration earlier. So what happens? Why this photorespiration occurs? Because of the oxygenase activity of Rubisco enzyme. We'll study about this photorespiration in detail in next session. But here just you have to remember that C4 cycle helps in the reduction of the photorespiration, which is a wasteful process. Okay, why it is called wasteful process? Because it doesn't produce any ATP. No ATP is produced here. Instead, ATP is utilized and carbon dioxide is produced during photorespiration. So it is known as a wasteful process. It also leads to the decrease in the rate of photosynthesis, you can say, or the photosynthetic yield of the plants. So in C4 plant, what happens? There is a reduction of the photorespiration by concentrating carbon dioxide around the Rubisco enzyme. You've already seen that in bundle sheath cells, Rubisco is present and carbon dioxide was released during the decarboxylation of malate, and this carbon dioxide enters the C3 cycle where Rubisco enzyme is involved. So C4 plants, what they do, they are able to concentrate the carbon dioxide around the Rubisco enzyme. Okay, so there is high concentration of carbon dioxide, less concentration of oxygen, and Rubisco binds to carbon dioxide and goes into the C3 cycle, and it gets fixed into the organic molecules such as glucose. Okay, so there is a reduction of the photorespiration which leads to the increase in the photosynthetic yield of the C4 plants. Okay, so this C4 cycle, it also enhances the ability of the C4 plants to thrive in high temperature and intense light conditions. So what happens in intense light conditions? The stomata remains closed. Okay, so that's why there is no loss of water, there is less transpiration, and the plant is able to sustain itself in high temperature and intense light conditions. So this was about the about the dark reactions of the photosynthesis, or you can say the dark phase, biosynthetic phase of the photosynthesis. And later in the next session, we will discuss about the rest of the cycles. Thank you very much.